Morning. I think we can start. Okay. So a pleasant morning to all of us. Welcome back to the second half of the two-day PSM webinar with the theme Microbiology from Concepts to Application Towards Sustainable Development. I am Noel G. Sabino and I will be your moderator for today, together with Dr. Marilyn Balolong. Okay. We're having this seminar via Zoom, but you can also watch the presentation via YouTube live stream. To formally start today's session, may I call on the president of the PSM, Professor Joel Cornista.
Good morning, everyone. Thank you very much for joining us again in our day 2 of the PSM webinar entitled Microbiology from Concepts to Application Towards Sustainable Development. Again, on behalf of the Board of Directors of the Philippine Society for Microbiology, I welcome all our members, esteemed guests, participants, sponsors, and partners to this webinar. Yesterday, we have heard from our invited speakers who are experts in their field of education and research, the role of microbiology and its allied fields in improving the lives of our people. Dr. Jocelyn Zarate talked on harnessing beneficial microorganisms as biofertilizers for sustainable development, while Professor Emeritus Dr. Milagros Gos lectured on fighting malnutrition, poverty, and global warming, gifts of algae to the world. Lastly, Engineer Ivan Henderson-Ge lectured on the processing integration of microalgal biofuel plant. Today, we're about to listen to equally relevant and interesting topics to be delivered by no other than Dr. Ronnie Kalugay, Dr. Grace Banaay, and Mr. June Barnes as our speakers for today's webinar. Today's program will be filled again with new learning and meaningful discussions about the role of microbiology and its allied fields in ensuring a sustainable future for everyone. I enjoin our participants to take this opportunity to contribute and make the most of this webinar. Again, I would like to thank our organizers, specifically Mr. Kristen Jordan O. De La Rosa, which is the head of the Cluster 2 and also the head of this webinar, Dr. Lucille C. Villegas, Dr. Noel Sabino, Mr. Robbie Vasquez, Dr. Donna Papa, Edison J. Pogoso, and of course, Mr. Bonnie Datul, who have worked hard to make this webinar possible. A special thanks to all our moderators, our host, Ma'am Marian, co-host, Ma'am Len and Ma'am Les, and our question takers are Howell, both in Zoom and also in our YouTube channel. I would like to extend again our gratitude to our partners, Procter & Gamble, for both Joy and Ariel. I'm very happy again to share to you new developments of a research in Japan, which actually showed that Joy is an effective surface disinfectant and can reduce viral count, which is a very good development, particularly now in the midst of a pandemic. We also like to thank ESCO Philippines. These are makers of biosafety cabinets. And of course, 3M Philippines, who leads in the manufacture of protective materials such as masks, and is represented by no other than Mr. June Barnes, who is also our speaker for today. Our partners have been with us through the years and have made all of these activities possible. Thank you very much to all of you. One of the mandates of PSM is to promote scientific knowledge in the field of microbiology, and we are committed to fulfill this mandate. So expect more webinars to come in the near future as we embrace the new normal in the midst of the pandemic. On a personal note, allow me to personally invite you to celebrate with us the 50th Annual Convention and Scientific Meeting next year. Hopefully, the situation will normalize soon and we'll be able to meet again face to face. I know that you are equally excited as I am, and let's just hope for the best. This pandemic has once again showed us how microbes, the very organism that we study, can affect and suddenly change our lives. Imagine going into the lockdown and not able to do the normal things that we do. But nevertheless, let us adapt and embrace the new normal so that this is the way to go in most of the conventions and symposium in the future. Okay. For the meantime, let us enjoy the rest of the morning's webinar in the comfort of our homes or in our offices. Stay safe and healthy, everyone, at mabuhay po tayong lahat. Thank you very much, and again, good morning to everyone. Sir Noel, naka-mute po kayo, Sir Noel.
Okay na po? Okay. So
Okay.
thank you very much, Professor Joel. Okay. Now, as mentioned by our president, we will be having a total, we have invited 3 distinguished speakers who will be discussing or presenting their research work. Okay, now please take note that we will be having an open forum that is after the presentation of all the speakers. So for your questions, okay, please post in the chat box for the Zoom participants. Now, as for those viewing, okay, the live stream on YouTube, you can make use of the comment section. Okay, now may I also request everyone to please turn off the Zoom participants, of course, to please turn off their videos and put their or your microphones on mute. Okay, thank you very much. Okay, so for our first speaker, okay, she is a professor at the Institute of Biological Sciences in UP Los Baños. She obtained her baccalaureate degree in biology, major in microbiology cum laude, her master's degree in microbiology magna cum laude, and her doctorate degree in microbiology summa cum laude from UP Los Baños. She has authored and co-authored numerous articles in scientific journals and presented a number, okay, of her papers in both international and local conferences. Okay, ladies and gentlemen, let us all welcome our first speaker, Dr. Charina Gracia B. Banaay.
Hello, everyone. Good morning. Thank you so much, Sir Noel, for your kind introduction. I'll just be sharing my slides. Okay. Okay, can you see it now? Okay. Okay, once again, good morning, everyone. And thank you, Sir Noel, for the kind introduction. It is my pleasure to be with all of you this morning, tell you about some ways by we as microbiologists can contribute to achieving the 2030 global agenda of the United Nations. This global agenda is really all about sustainable development. And this presentation seeks to connections between our work as microbiologists and the concern of sustainable use of soil resources for the sustainable development of communities. So this presentation is all about soil, society, and society. Rooting for Earth is about harnessing soil microbial resources to support sustainability. To you a quote from the late President Franklin Roosevelt in a letter to all state governors on a uniform soil conservation law. He wrote the following. The slide is not moving. Okay. A nation that destroys its soils destroys itself. So that was written in February 1930s, the Dust Bowl of the 1930s, which is caused by excessive soil erosion and drought. So clearly, soil and sustainable societies are intimately related. If the soil is impoverished, crops will fail, and society will be left without food. Of course, economic devastation will ensue. The principle goes like this. Healthy soil begets healthy crops, and healthy crops beget healthy products, and of course, healthy people. Now, we're not just talking about a healthy environment, but a healthful environment. And by healthful, we mean that it's not just healthy by itself, but it also promotes health for all things that depend on it. The products, of course, are not just food for humans, but also feed, fiber, and fuel. So clearly, really, healthy soil can be considered as one of the cornerstones of life. It supports biodiversity, food production, confers water benefits, and contributes to carbon storage. The last one is a property that makes it important in climate adaptive systems. But what is the foundation for healthy soil? What makes soil healthy? Said that a vibrant, healthy soil arises not from soil itself, but from the interactions of plants, microbes, and animals in the soil environment. In other words, soil cannot be healthy by itself. It is the living substrate of soils and plants and interactions that is sustaining and driving soil qualities. It is that living substructure that is key to making soil healthy. It's not just the structure, but the structure of these organisms, but more importantly, the interactions between them that make soil healthy. And if we're talking about interactions, that's actually ecology. So, what comprises this living substructure? What makes up life in the soil? Yeah. So, soil contains organic matter, and 5% of the organic matter content of soil is actually soil organisms. And of the soil organisms, majority, it's estimated that it's around 80%, is composed of microorganisms. So a majority community consists of microorganisms, of which only a fraction are culturable. It has been estimated before that in the bulk soil, only around 1% of microorganisms are culturable. Here, soil, not the bulk soil, but in rhizosphere soil, more than 50% are culturable, and that is good. We can isolate them and a well-known microbiologist once said, our soil teems with a multitude of organisms provide the necessary work for healthy plants to grow free from disease, pests, and infertility. Clearly, Microorganisms have a vital role in sustainability of societies. Let's now take a closer look at microorganisms associated with plants. We call them the plant microbiome. This refers to all the microorganisms living in, on, or near the plant. It includes those found in the air surrounding the plant, on its leaves, the phyllosphere, inside its body, the endophytes, and on and around the roots and in the bulk soil. Let's zero in on the root soil interface. There are three distinct zones in this interface. There's the root endosphere, referring to the area inside the roots, the rhizoplane, which is the surface of the roots, and then there's the rhizosphere. The rhizosphere is described as the soil immediately surrounding and directly under the surface of the plant roots. So if you can see this picture, you have an idea that individual roots are actually overlapping. Lawrence Hiltner, soil microbiologist and pioneer in rhizosphere microbial ecology research, coined the term rhizosphere in 1904. And he described it as an area that is constantly in a state of dynamic flux because of the root exudates that constantly change, affecting the properties of the soil as well as the microorganisms and their interactions. In other words, the rhizosphere is a highly connected and interactive system. This zone is characterized by intense biological activity owing to the release of the root exudates to stimulate or inhibit rhizosphere organisms. Let's take a closer look at the root microbiome, biological activity that is described as intense, no, because of the root exudates. So the root microbiome consists of around 10 to the 8 to 10 to the ninth per gram, consisting of more than 10,000 species. The rhizosphere microbiome in particular is said to extend the functional repertoire of the plant. Oh, how does it do that? How does it extend the functional repertoire of the plant? It does this by mediating plant soil feedback. How does it mediate this? No, it is able to influence growth of the plant, mediate nutrient uptake, confer host protection, and even influence phenology, the timing of events in plants. So the rhizosphere microbiome extends the functional repertoire of the plant by affecting the plant phenome. What do we mean by phenome? This is the set of all phenotypes expressed by the plant. So oftentimes, the root microbiome, the effect of the rhizosphere microbiome is described as the plant's second genome. How is that? Because it affects the phenotype of the plant. That's why. But it all really starts with the plant, the host plant. Depending on various host factors such as the host genotype, developmental stage, and the health and fitness status of the plant. Now the plant sends out signals in the form of root exudates here. you know, the below ground properties. It sends out signals in the form of root exudates, and this affects the kind of microorganisms in the rhizosphere, which in turn affect the phenome of the plant. It's a type of feedback mechanism, as you can see here, and the plant and its microbiome adjusts with each other's presence and their environment. But the point is, it all starts with the plant. And it starts with the root exudates. Now, the root exudates would be part of the rhizodeposits. The microorganisms and other fauna thrive on the rhizodeposits, which form the base of the rhizosphere food web and the rhizosphere interaction. Now, so you can see here very active um, interrelationships and interactions between these factors, the plant, the environment, and the microorganisms and other microfauna and the soil. Now, so you see here a very dynamic crosstalk between the plant and the rhizosphere microorganisms in relation to its environment, various environmental stimuli, the soil properties, and the effects of the whole microenvironment. In simple terms, the rhizosphere can be described as a continuously evolving habitat which influences and is influenced by interacting biotic and abiotic components. It is these dynamic interactions that form the basis for the importance of rhizosphere ecology in achieving the 2030 global agenda. There are 17 sustainable development goals, as you can see. I believe microbiologists can contribute to all 17 goals. Even as individuals, we can support all these goals in our own way. In relation to my presentation, let us concentrate on 6 of these goals, namely and well-being, sustainable citizen communities, responsible consumption and production, climate action, and life on land. Let's start with zero hunger. Zero hunger. The challenge really is quite daunting. This means that there should be double the production of food, double the production, or reduce at least into half the amount of food wasted in order to feed 9 billion people by 2050. That's just 30 years from now. So really it's all about food security. And in the context of rhizosphere ecology, this means crop productivity. It involves soil fertility, nutrients, water, stress response, to name a few. I have listed there 6 benefits that rhizosphere microorganisms can contribute to increased crop productivity. Okay, so there's nutrient cycling and nutrient uptake by plants. This is actually part of the normal functional properties of microorganisms in the ecosystem. They do this naturally. So all to do is really to harness that property. Disease suppression. No, rhizosphere do this by many different means. One could do it by direct antagonism, either physically, mechanically, or chemically. You know, do it by competition or antibiosis or some other means. The last 3, plant growth promotion, plant defense response, and abundance tolerance, are quite complex processes. They involve a lot of factors. And importantly, they involve of signals between the microorganism and the plant. The microorganism affects the plant metabolism and therefore affects its phenotype. All of these 6 benefits are based on this. They're based on the signaling network within the rhizosphere, a crosstalk between the plant and the microorganisms in relation to the stimuli that is present in their environment. And these signaling networks are based on the fact that plants have co-evolved with microbes, genotypes that support microbes that increase health of plants. Like this interaction, we shouldn't set this aside because this is an evolutionary interaction. The interaction has developed through time. Although we already know some aspects of these interactions, there is still much to learn. We need to dive into the mechanisms by which these benefits are carried out. There is much to do because each plant soil microbe system is unique. So we expect differences despite possible core themes. The second one, Okay, good health and well-being. When we talk about good health and well-being, it is necessary that we think of it as one health. Because we recognize that everything is interconnected. There are existing ecologies here that we cannot set aside. One health is the approach to designing and implementing programs, policies, legislation, and research in which multiple sectors communicate and together to achieve better public health outcomes. This means that in order to have good public health outcomes, we necessarily have to include actions toward good environmental health and food safety. Again, we go back to the principle that healthy soil begets healthy crops and products. So rhizosphere microorganisms help contribute to good health and well-being through nutritional improvement of food and feeds. How do they do this? Through all of their activities, growth promotion, bioremediation, biological control, stress tolerance, and other properties. I want to emphasize this nutritional improvement of food and feeds. There is few research conducted here in the Philippines regarding the ability of beneficial microbes to improve the human health promoting properties of crops and their products. True, we look at agronomic properties. They grow fast, they grow big, there's high productivity. But we also have to look at the health promoting properties of the food and feed products themselves. Research has shown that beneficial microbes are able to promote and enhance the ability of host plants to produce certain phytochemicals that are healthful. We need more research that sheds light on the ability of rhizosphere microbes to increase the health promoting properties of crops, especially food plants. Traditionally, when we want to improve crops, we resort to plant breeding. However, crop breeding has largely decoupled, it has separated the rhizosphere microbiome from plant selection. If important interactions have been lost as a result, there is actually an opportunity for us to re-engineer characteristics of beneficial rhizosphere microbiomes back into agricultural cropping. So that's rhizosphere engineering. Some of you might be interested in this. Designing the rhizosphere by harnessing beneficial plant microbe interactions and other rhizosphere activities. There are two ways of doing this. One side is the plant, the other one is the microbiome. So you can redesign the plant so that it can give out correct and stronger signals, the root exudates, to cause optimal microbiome assembly. On the other hand, you can engineer the microbiomes, like you assist in the correct assembly of the microbiome by adding to the rhizosphere. Now, we can discuss that another time. The next one is sustainable cities and communities. So sustainability is meeting the needs of the present without jeopardizing the ability of future generations to meet their own needs. It rests on three pillars: the environment, the society, and the economy. In other words, it's planet, people, and profits. In the context of a rapidly urbanizing area, this means building and managing urban spaces. In the rhizosphere ecology in particular, this means creating and optimizing the green spaces that are available in small areas. That's the problem here in the Philippines. No? When we have urban planning, we don't include green spaces, and green spaces are very important. No, so because we only have very few or small areas of green spaces, we need to make it efficient. And that involves making them, the functions of those green spaces, optimal, or at least near optimal. So that includes optimal phenome of the plant. And the rhizosphere microbiomes can contribute to that by increasing productivity, increasing resistance, and other properties. The optimal functioning of that green space, that would mean for rhizosphere microorganisms an efficient nutrient cycling process, remediation if necessary, biocontrol, and other properties. Okay, so for optimal functioning, all of these involve core microbiome assembly. No, more core microbiome assembly for optimal network communication and function. What do we mean by this? Research has shown that plants initiate communication with soil microorganisms by releasing root exudates. And if the initial set of microorganisms assembled at the start are the essential core members, a highly connected and robust network of microbes ensues. This is called the priority effect. I'll give you an example. Consider this data here as obtained and analyzed, showing the network of connections between rhizosphere is one species, and the lines represent their connections to other species. Some of these species are more others, as you can see here. So this one is highly connected, this one is not. We know that it's an essential species if it affects network stability greatly. Take this blue circle, this species blue. If we take it out here, we know that it's essential if the rest of the network will collapse like this one. Yes, it's essential if the network collapse, or if it doesn't, like this one, the network is maintained, that it is not non-essential. Core rhizosphere microbes are always essential, especially at the start, at the start of the assembly of the rhizome microbiome. That's called the priority effect. So without them, the core microbiome, the efficient connections will not happen later. Okay, so for optimal functioning, we need to have at the start, at the onset, the core rhizome microbiome assembly. Okay, moving on. Number 12, responsible consumption and production. This goal is actually all about reducing our ecological footprint by changing the way we produce and consume goods and resources. Let's focus on various production systems: organic agriculture, agroforestry, conservation agriculture, integrated forest farm, and fish systems. Harnessing their microbial ecologies are part and parcel of all of these production systems, as they capitalize on existing ecologies to support crop production. Their overall goal, the negative culture on the environment, to reduce the ecological footprint. Next one is climate action. This is all about actions to combat climate change and its impacts. Rhizosphere ecology is compatible and consistent with the goals of climate smart agriculture, which is based on 3 national food security and development goals, namely increase in productivity and income, reducing agriculture's contribution to climate change, and strengthening resilience to climate change and variability. If we break these 3 down into specific goals, we'll end up with 6 targets. The first 5 here, can be supported by rhizosphere ecology. Increasing productivity. This is where the growth promotions, biocontrol, disease suppression, resistance, and tolerances come in. They can increase overall productivity of crops. Increase net return. If you have less inputs due to action of microorganisms, there's greater return for the farmer. Improve input use efficiency. There's higher input use efficiency because you need less inputs to get bigger returns on product, because you're harnessing the beneficial actions of microorganisms. There's also reduction in emissions as we design for long-term carbon storage. How do rhizosphere microorganisms do that? They improve carbon storage through increasing plant productivity. Now, because the plant is acidic, so if you increase productivity, then you also increase carbon storage. You also decrease microbial respiratory carbon loss because the interaction the connections within the rhizosphere is more tight. And as you increase diversity, nutrient cycling becomes more efficient and it happens in a closer system. So you have less CO2 emissions. There's also increased resilience because the plants are more tolerant or resistant to both biotic and abiotic stresses. Well, for CSA, climate smart agriculture, to happen, we really have a lot of work to do because climate smart interventions are highly location specific and knowledge intensive. Each plant soil microbe system that you want to look into is unique. You really cannot just adapt interventions from another system. You have to make it optimal for that particular system. So considerable efforts are required to develop the knowledge and capacities to make CSA a reality. Life on land. The essence of rhizosphere microbial ecology is really to understand and harness soil biodiversity and interactions for enhancing its beneficial effects. It involves utilization of this largely unknown natural resource to support and strengthen ecosystem goods and services. Okay, I only have a few minutes left. I'd like to go into some areas of research you might be interested in this. So I'll just give a brief outline of what we can do. Okay, the first one is, of course, we have to know who's there and what they're doing. So look into research on root microbiome structure and function. This is where molecular tools can come in really handy. I have listed down through four metas, metagenomics, metatranscriptomics, metaprotein metabolomics. This would help tease out uh, which microorganisms are there and different stages of developmental stages. What are they doing there? What are their possibilities or potentials? And that's already a lot of work. After that, you don't stop with um, culture independent methods. You proceed with looking at the rhizosphere ecology because that's the meat of everything. It's the action. So we look into that. And by doing that, we need to combine culture independent with culture dependent techniques. The basic search towards this direction will start with phenome based sampling of the microbiome. Subsequent scrutiny to address how the functional microbiome is shaped and how it connects to the plant host phenome. How do specific plant root extract shape the microbiome to its advantage? You get insights on plant cues and traits that sculpt the root microbiome and how plants recruit their microbiome to maximize both nutrition and defense under often harsh environmental conditions. Remember, it starts with the plant. So, what are these signals? The plant is able to recruit the functional microbiome. SynComs, these are synthetic communities. Others call it MEMS, minimal effective microbiome set. Some even call it agricultural probiotics. So in other words, it's a microbial consortium, a group of microorganisms that you apply to the plant, especially during the start, at the onset, while it's still a seedling. What do you put in there? The minimum effective microorganisms that will confer a greater advantage on the plant. There are many lines of research you can do here. You can look at comparison of microbiomes of modern and ancestral varieties. You can look at specific concoction, a combination of these minimal effective microbiome set, and see how the plant affects. Now, returning to culture-dependent microbial surveys allows the construction of experimental ecological systems so that we can understand interaction. And plant microbiome vault. What is this? So we are used to having culture collections that have pure cultures, but this time you are using a specific plant phenome, whatever plant phenome that you have in mind, and then you extract the whole rhizome. This is what you keep in the vault for future applications. No, it's like this. No, you start with phenome-based sampling. You isolate the microorganisms there. Side by side, you do culture-independent profiling, and you use information from these first two steps to inform construction of synthetic communities. And then you put it all back into the experimental plant and see if you can derive the optimal phenome that you are after. Okay, so in summary, determining rhizosphere microbiome structure and function will provide insights about the plant-soil microbe interactions. And this will help us harness the power of microbes to increase crop health and productivity and promote sustainable development. Thank you very much for listening.
Okay. Thank you very much, Dr. Grace Banay, for that very wonderful talk. So please reserve your questions for Dr. Banay. You can type your questions in our chat box. No? So we have Donna and Howell who will be collecting all your questions. And then we will all be throwing them later to our speakers. Okay. All right. So I think we will move on to our next guest speaker. Okay. So our next speaker is a faculty member of the University of the Philippines, Baguio. Of course, I'm sure everybody knows our next guest speaker. No? He has been serving the Philippine Society for Microbiology for years, and I'm sure he's a friend for everyone. May I introduce to you our next speaker? He will be talking about lights bacteria action. It's medyo showbiz talaga to. No? So let's please give a round of applause to Dr. Ronnie Kalugay.
Okay, thank you so much, Len, for that beautiful introduction. And it's really a pleasure and also a pressure to be online for the first time because now we're being watched, I think, around the world because we're being watched on YouTube too. So good morning, everyone, to the members and all watching. Okay, so today I'll be talking about, is my screen on? Okay, bioluminescence, and I entitled my talk Lights Bacteria Action. So like what Ma'am Len said, it's very showbiz. But actually no, every time, have you seen bioluminescence in the wild? So if you have, then once you see them, it's really a spectacle. It's like a scene in a fantasy movie or in a science fiction. But actually, no. For the microorganisms that engage in, that have bioluminescence, it's actually just another biological function. And that I'm going to talk more in detail, okay, and give you some examples, okay. So let's go light, okay. So it's a glowing world actually. So there are so many bioluminescent organisms across taxa. So it's not just the firefly. Actually, 80% of bioluminescent organisms are in the vast ocean, okay. And you can also find them in terrestrial and even underground. Okay, there are bioluminescent fungi. You don't see them because they're underground, but scientists know that they are there, they exist. Okay, so let's talk about this light. So this light, okay, oops, my slide is not moving. Yeah. So bioluminescence is the emission of light from a living organism. Okay. So the most common organism that we know are the fireflies. Okay. So like I said, for us, it's an entertainment. So if you want to witness firsthand bioluminescence, you go to Palawan or Bohol, and they have what you call the firefly tour. So they take you to an island, and then this is what you see. You see an island full of trees, and the trees are festooned. Like it's like festooned with Christmas lights. It's like Christmas during summer. This is what you will see. Okay. Yeah. So again, it is not in detail that's entertainment. It's actually very scientific biological function why they emit light. So one of the most common biological function actually is for mating. Okay, like your fireflies. Okay, they try to attract the females, try to attract the males. Okay, and then they start the courtship and they mate. So and then they multiply in population. Okay, and the most common function also for bioluminescence is for predation actually. Okay, so yung firefly din, hindi lang pala for mating ang paggamit niya ng pag-emit ng light. There is this genus Futuris, the female. Okay, it emits light to attract other species of fireflies. Yun pala, it's not going to mate with the male, which she attracted. But once the male comes, and then she will devour him. So it's like the black widow. So nandadaya siya. She tries to imitate the light communication signal of other species so that it can lure the other males, and then it will devour it. Okay. Okay. This is a cave in New Zealand, and the ceiling of the cave is there are hanging threads of bioluminescent mucus-like substance secreted by a type of a gnat. Okay, so actually this is not also you can see the humans are looking up again. It's a tourist spot. But actually, for the bioluminescent organisms, this is a type of predation. They're actually other insects living in that cave. Once they come out from their eggs, or when they wake up in the morning, when they look up, they think that they are looking at the sky. So they think they are looking at the stars. So yun ang theory. So what ang tendency nila, they will fly upward. And once they fly upward, they get trapped on the sticky bioluminescent threads, and then the larvae, they start to feed on them. Okay. Yeah. Again, the other function is for defense. So medyo may mga organisms na hindi pa clear kung defense nga ba talaga ito, and we will know why later on. So defense against predators, of course. And so before I give an example of that, have you seen bioluminescent beach water? Yeah, these are actually your dinoflagellates. Okay, bioluminescent dinoflagellates. They're just floating in the waters. And they are touch activated. Kaya sila umiilaw because when the tide comes, when the wave comes, then they are activated to emit light. And believe me, I've seen this in many places in the Philippines. Okay, and of course you don't see them. Maybe most of the time you go swimming at daytime. But ako, ever since I've engaged in this research, I make it a point every time I travel that I go out and shake the waters. Okay, and ganyan nangyayari. Okay, floating dinoflagellates. Okay, ito wave. Okay, and ito, this is captured in California. Nagsusurf siya. And they are dinoflagellate bloom. Okay. Yeah. So and this is the under the microscope, one type of a dinoflagellate, Noctiluca, which emits light. Okay. So, bakit tong mga microorganisms na to nag-i-emit ng light? Ano ang function nila? Okay. And the theory is, for this function, they call it the burglar alarm theory. The burglar alarm, meaning if the thief comes, there is an alarm. Meaning, in this case, this is how they call them. But in this case, when a predator comes, the predator of the predator is being eaten. Okay? At yung, how will I explain it? Okay? Let's watch a video. Okay? So this is David Attenborough, the pioneer in science journalism. Okay? This is an example of a burglar alarm. Okay? So, itong hipon na ito, it's walking on dinoflagellates. Okay? And once, kasi touch activated siya, once naglalakad siya doon, nakikita siya nung predator niya yon. Kinain siya nung squid. Okay? Kasi ang advantage niyan ay doon sa mga dinoflagellates. Ay nako, kakainin na naman tayo ng hipon. Pero ay nako, mag-signal na tayo na nandyan na si hipon at itong si hipon makikita siya ni octopus at kakainin niya yung hipon. Okay? Clear ba yun? Yun yung burglar alarm theory. Okay? There's another example. Yeah, ito. Yung the crab preys on the brittle star. Okay? So, the adaptive mechanism to evade the crab by the brittle star is to, once the crab touches the brittle star will start to emit light. And that emission of light will be visible to the predator of the crab. So instead, before, pag nakita makikita noong predator ng crab na umiilaw at magiging visible yung crab at kakainin na siya ng predator niya. Okay? So here, an example. Yan. Ito yung arms ng brittle star. And here comes the crab. That's the arm of the brittle star. It's hiding under the rocks. Watch. Ito na yung crab, dahan-dahan siya. And the octopus, which is the predator of the crab, is watching nearby. Okay. Ayan na. The crab has touched the brittle star and it emitted light. And this light emission was seen by the octopus. And ayan, here comes the octopus and it ate the crab. So before the crab can eat the brittle star, the crab was eaten by the octopus. Kasi mas prefer ng octopus yung crab. Pati tayo, di ba? Mas gusto natin kayo. Ayan, this is a diver which touched the brittle star. So it's also touch activated or it responds. It's a survival response. Okay. Okay, so this is another example. These are uh, another type of planktonic microorganism, and this fish is actually invisible underwater yun. Pero once na swinallow niya yung nag-float na plankton na yon, pati siya maglo-glow tuloy at nagiging visible na siya sa predator niya, pero bigla yang niluwa yan, hindi na naman siya makita ng predator niya. Yan. So that's one. So parang superpower siya, but it has a clear biological function. Pero, and another, so those are the functions, okay? So recently, I'd just like to add a beautiful discovery that may mga turtles na bioluminescent sila. Yung mga pawikan, nakita nila somewhere in South America. But it's not actually the carapace itself na nag-emit ng light. It's actually the microalgae which are living on the carapace of the turtle. So it's a kind of symbiotic relationship. Okay? Kaya naglo-glow yung turtle at night. Okay. Ayan. So ito rin naman, this is what you call the firefly squid. And it is a delicacy in Japan for hundreds of years. Okay, so to give you an example, this is another example of to evade predation. They have yung pag-emit ng light nila ay through photophores, and when they surface to shallower waters, they change color. Yung photophores parang they turn into green. So they tend to blend with the color of the water. At kung marami rin kayo at umiilaw kayo, nagmumukha kayong malaki na organism. So matatakot yung mas malaking. So this is in Japan. This is how they harvest them, umiilaw. Okay. So all this I mentioned are, they emit light through their biochemical function on their skin or within them. So let's go to bacteria which glow, okay? That glow, okay? So bioluminescent bacteria was first discovered in a nocturnal squid that glows. So mayroong squid na paggabi, gising siya and it emits light, okay? And that emission of the squid, unlike yung firefly squid na pinakita ko kanina, the light coming from this nocturnal squid is actually coming from symbiotic bacteria inside its light organ. Okay. Yan. And this is the Euprymna scolopes, or the Hawaiian bobtail squid. So maliit lang yan. You can see that's a figure. It's 1 centimeter. It's a very small squid, but that's already an adult. Okay. So this squid, ito yung symbiotic bacteria niya, yung Vibrio fischeri, okay? And it lives inside, it evolved to have a structure called a light organ at doon nakatira yung mga Vibrio fischeri, okay? So, paano yan, ito may pag-aaral silang ginawa. The light organ is actually located at the ventral part of the squid. Okay, so bakit ventral, malalaman natin. I'll tell you all about that. And then they had this type of special microscopy. The blue are yung ito siya. I-zoom natin yan, and ito siya. Okay, they recruit the Vibrio fischeri through this way. So these structures with the mucus, it serves like a sieve for bacteria because there are so many other bacteria in their environment in so many sizes. Pero the big question is, Vibrio fischeri lang ba ang nandoon sa light organ na yan? And the answer seems to be yes. Okay, it's pointing to that. Pero paano nila ginagawa yon? And they found out that this light organ, the squid, releases chemicals which are specific for the Vibrio fischeri to be attracted, and they enter this hole into the light organ. So it's a chemical communication between the squid and the Vibrio fischeri, in which the Vibrio fischeri are chemotactically attracted into this light organ. Okay. So I guess the next thing magandang aralin that I should do next is do microbiome studies. Okay. Oops. So, ito yung yung in captivity, yung Hawaiian bobtail squid, maliit lang siya. It's nocturnal. Okay. So during the day, it burrows under the sand. Okay. Ayan siya. Tulog, gusto niyang matulog. So yan. It will burrow under the sand, and then it uses its tentacles to to cover itself. Okay. So cute. Yan. And then at night, it will wake up, and then it will emit light while it's grazing for food. So yan, hindi mo siya makikita. So that's another defense mechanism. It has to hide itself from predators. And then at night, it will prey upon other. Okay. So, so at night, makikita nyo dyan na yan. So at night, umiilaw yung mga bacteria. So ganyan siya. Nasa ventral part yung light organ. So magdi-diffuse yung light na ganyan paggabi under the moonlight. At yung view na yan, this is the perspective of its predator dito sa ventral part, because it's usually in shallow water. So meron siyang invisibility. It seems like may invisibility power siya, but it's all just biology. Okay, yeah. Okay? So yan. Hanggang hindi mo na siya nakita once nag-light up yung mga Vibrio fischeri na symbiotic bacteria niya. Okay? Yan. Okay. So, they studied this Vibrio fischeri. How do they emit light? So, these microorganisms emit light when they are only at high cell density. Meaning, pag marami sila, they emit light. Pag konti sila, they will not emit light. So, and why is this so? Okay. Yan, that's so, ito, gising, at ito, nag-emit na ng light. Just a repetition. So, that cell density dependent biological function is actually called quorum sensing. From the word quorum, di ba? Pag nag-meeting kayo, may quorum na ba tayo? Ganon. But meaning, it's a specific number of members which can launch a function. That's what you call quorum. And sensing means they can sense kung marami sila. Okay? And hence, they can launch a function. And in this case, it's a bioluminescence. Okay? And yun ang advantage nun. So anong advantage ng bacteria? It has a rich life inside the squid. Okay? Because once they're out in the vast ocean, nutrients are more limiting. But once you are in a structure which the squid has developed over time, nutrients are always rich there. Okay. Yeah. So, pag nagigising yung squid ng gabi, and then magre-graze siya, sinasabi ba niya, uy, gumising na kayo mga bakterya, umilaw na kayo dahil gising na ako. May nangyayari bang ganon? Actually, it's a very elegant design of nature. And we know that bacteria, after 16 hours, they reach logarithmic phase. Okay? Meaning, high cell density na sila. Let's review. At low cell density, no light. At high cell density, there is light. So, habang tulog siya, while it's buried in the sand, the bacteria are low in population. At if low in population, low cell density, walang light. Okay? Pero as the day goes on, pagdating sa gabi, they reach a certain population na high cell density sila. At that time, gising na rin yung nocturnal squid, so iilaw na sila. Okay? And the squid uses that light for counter shading to evade predation. Okay? And then, pagpababa, when dawn comes, mababa na yung population, low cell density, no light. Okay? We know that bacteria, they only have around 24-hour growth curve. Okay? And some of the dead cells are being squirted out by the squid. And so a few cells are only left. And the cycle goes on day in and day out. Very elegant design of nature. Okay. Yeah. Low cell density, high, no light, high cell density, there is bioluminescence. Okay? Paano nangyayari yan? Because of these auto-inducers na yan. These are the molecules which trigger a cascade of reactions and gene expressions, and at the end, they emit light. Itong konting red dots na to, kulang yung concentration na yan. Pero pag marami sila, yung mga red dots na yan, yung auto-inducers, they meet the threshold of concentration enough to trigger the cascade of reactions to express bioluminescence. Okay, so it's amazing because may mga genes pala na eto. May mga genes pala na they are only expressed pag marami kayo. Pag mag-isa mo, you cannot express that gene. That gene will not be upregulated. It will not be expressed. Pero pag marami kayo, na-express siya. Amazing. Okay? So itong signal na to, this is the autoinducer. So it has a specific receptor. And when it triggers the DNA to be expressed, a set of genes, which are the lock genes. And then, ito na yung expression. In this case, it's the bioluminescence. Okay? So, mamaya, napaka-importante nito. May kinalaman ito sa burger, fried chicken, yung mga fast food na kinakain natin. Okay? So, I'll go fast. Okay? So, bacterial communication through chemical signals also is another definition of quorum sensing. Okay? This is the language of bacteria. Okay? It's also called quorum sensing. How did this happen? Okay? So ito yung signal, yung autoinducer. Again, it has a specific receptor, and then it triggers the DNA to be expressed. Okay? Remember that. Okay. So ito yung mga microorganisms, bacteria, which have engage in quorum sensing. Okay? They have what you call autoinducer 1. So ito, yung autoinducer ng members of this genus. Okay? Ito naman, ito. So like I said, each signal has a specific receptor. So this signal cannot enter the receptor of the signals here, of the receptors here. This signal cannot enter the receptor here, or this cannot enter the receptor here. So specific sila. So yung mga members lang nito ang pwedeng mag-admit ng mga signal na ganito because they have the receptors. Okay? So that in effect is a chemical communication. Kasi pag once na pumasok yan, meron na silang action together as a group. Okay? So ito yung Ilocano, ito yung Pampangueño, ito yung Pangasinan, ito yung Cebuano, ito yung Boholano. Okay? Yan. Sila lang nagkakaintindihan. Okay? Yan. Pero kung titignan yung mabuti, yung chemical structure, meron silang common structure. Okay? Na yan. At itong common structure na yan, they also excrete that, and it is called the autoinducer 2. Yung una is autoinducer 1, pero itong common na to is autoinducer 2. yan na yung Tagalog, yung ginagamit natin as a common language. Sa kanila, yan yon, yung auto-inducer 2. Kasi in nature, they do not exist as exclusively sila-sila lang. Okay? Pero they are a mixture of populations. And so they need a signal communication molecule, and that is called your auto-inducer 2, or what you call a universal communication molecule. Okay? So, in effect, meron silang, not effect, meron silang dalawang type of receptors. Okay? Ito yung first language nila, yung dialect, and ito yung Tagalog. Okay? Because in a mixed population, they also there is also synergism, right? So and all these are expressed through a group behavior genes, lalo na to. Okay. Yeah. Okay. So quorum sensing accomplishes a task and reaps benefits as a group, but you cannot attain it when you are alone. Okay? Yung lux genes na yan, hindi mo pwedeng i-express, hindi ka pwedeng mag-shine mag-isa mo. Kailangan mo ng dapat marami kayo. Okay? Kasi nga, kulang yung auto-inducers to trigger the expression. Okay? So lux genes are actually called the group behavior genes. Amazing. Okay. Okay, let's go to action. Okay. What can we benefit from this basic information of bacterial quorum sensing and bioluminescence? Okay. One of the pressing problems now in the world is the antimicrobial resistance. This graph noong 1950, we only have Staphylococcus aureus, which are resistant to antibiotics. But as through history, we've been using antibiotics. The other pathogens mutate, okay? Because that's nature. If you apply selective pressure, just like applying antibiotics, the microorganisms survive and mutate. So over time, parami ng parami ang mga pathogenic antibiotic resistant. Okay, so this arrow just means na it's ongoing. The number of antibiotic-resistant pathogens and bacteria are increasing because of antibiotics, the continuous use of antibiotics. Okay, so anong gagawin natin? Right now, that's what we do, right? When we're infected with bacteria or other pathogens, we do need to take antibiotics. Okay. Okay, before we go, I'd like you are what you eat. Meaning, ito na yung sinasabi kong ano ang connection ng umiilaw na pusit sa kinakain nating mga fast food. Okay, you are what you eat. Meaning, do you know that what you eat can give you genetic and phenotypic traits? Okay, and it's been proven by science. Okay. So this is because of a phenomenon called conjugation. So this is your plasmid, okay? And this plasmid carry the genes for antibiotic resistance. And we know that there is bacteria conjugate, and when they conjugate, okay, this plasmid replicates and transfers the genes to the next bacterium, okay? So what was a non-resistant bacterium, after conjugation, it has become also an antibiotic-resistant bacterium. Okay, and this is what you call also horizontal transfer. Okay, yan yung plasmid. We all know that. So one classic example, bakit how genes are transferred from what you eat. Itong Bacterius plebeus, meron na siyang gene na for the expression of porphyrinase. Okay? Itong enzyme na to is expressed in the marine environment by the bacterium Sobelia. Okay? Paano napunta itong gene na ito sa gut bacterium ng tao mula sa dagat napunta sa lupa? Okay? So it is because of the culture of what we eat. Okay. Yeah. This enzyme is degrades the seaweeds. Okay. And these bacteria are attached on this seaweed and they degrade them. But over time, humans have been eating this seaweed for thousands of years. Okay? And so, na-transfer yung gene na yan sa ating human gut because of conjugation. Okay? At sino ang kulturang kumakain ng seaweeds? Okay? Ang mga Japanese. Isa sila dyan. Okay? I know for a fact because I stayed there. So, they eat seaweeds. They in and day out. Lahat ng meals may seaweeds. Kahit yung soup, may seaweed. Okay? Pag may party, may seaweed. May nori, may maki. Ganyan. Pero masarap siya. Okay? So, ano naman yung kultura na hindi kumakain ng seaweed are the Americans. So they did not find that gene expressing porphyrinase among Americans because they don't eat that much seaweed. So this study was done by nature. So that's one classic example of you are what you eat. It can give you traits. And now this population, the Japanese population, can degrade seaweeds. They have that ability because they have acquired that gene from what they eat. Okay. So ngayon, usong-uso ang fast food. Okay. Especially now, we eat delivery food. Okay, and fast food siya. So ito ang mahilig tayo, manok, fried chicken. Okay. So ang mga chicken, ito na ang modern existence nila because of our modern way of consuming food. Okay. Have you ever wondered when you go to the supermarket and you see free range nakalagay? And it's much more expensive than yung parang generic brands from the big supermarkets. Because yung mga generic brands, they are cultured this way. Hindi na tulad sa probinsya na yung di ba sinasabi natin, pag native yung manok, masarap siya. Ganyan. So mahal ang, now you know why mahal yung free range. Kasi it's organic, it's not fed with antibiotics. This chicken, they have to add antibiotics into their feeds. Kasi pag nagkasakit ito, mahawaan niya yung iba, so wala na. Magkakasakit silang lahat. So, yung wala na tayong chicken, konting plug, there was a time na ang headline is, today is a chicken sad day kasi hindi nakapag-serve ang Jollibee ng Chicken Joy. Kasi nagkaroon ng culling of chicken, nagkaroon ng shortage. So, pag ikaw ang proprietor nito, you have to put antibiotics so that you can save them from infection at hindi sila magkahawa-hawa at hindi bumagsak ang business mo. Okay? But over time, when we keep on feeding antibiotics to this chicken, yung mga bacteria nila nagiging antibiotic resistant. And when we eat this chicken na hilaw, uncooked, and nagkataon na na-ingest mo yung bacteria na yon na may antibiotic resistant gene, then that gene can be passed on to you and then you become antibiotic resistant. There are individuals who are allergic to chicken, but actually you check again, maybe you are allergic to the antibiotic residue but not to the chicken. So ang gawin mo, kumain ka ng free range, try mo kung magkaka-allergic reaction ka o yung native chicken. Kung wala kang allergic reaction, it's the antibiotic residue, not the chicken. Okay? So this diagram illustrates it very well. Antibiotics fed to our livestock, and we eat the livestock. So if you don't cook it well, especially when you grill, mga ganon, so you can ingest uncooked bacteria and they can proliferate in your system and they have those antibiotic resistant genes. Okay. So the other way we fight pathogens are through vaccines. Okay. But we all know about that. Okay. And I just hope and pray that we will have a vaccine for this pandemic soon. Okay. But I'll jump right away to what I explained about quorum sensing. Okay. So we need novel antibiotic scheme or alternative to fight this global menace of antibiotic resistance. How? Okay. Enter bioluminescent bacteria through quorum sensing. And anti-quorum sensing is also called quorum quenching. How is this done? Okay. At low, again, let's review. At low cell density, there is no light. At high cell density, there is light. Okay. And lo and behold, that conversation is, that bacterial chemical communication is also adapted by pathogens. When they are low in number, they are non-pathogenic. And when they are high in number, their autoinducers are enough to trigger a cascade of reactions to express virulent factors. Okay? So yun pala, they also engage in quorum sensing in order for them to be pathogenic. Okay, so so that knowledge on quorum sensing of bioluminescent bacteria led to this discovery. Okay, so in general, your bioluminescence expression are expressed by your non-pathogenic bacteria, but your pathogenic bacteria express quorum sensing through biofilms like your Pseudomonas aeruginosa. This is very deadly. It makes a thick biofilm in your lungs, and that's your lungs. Then you can die. Okay, and when the pathogenic biofilm is so thick, tumatalsik yung antibiotics. So nagiging resistant siya not only genetically but physically. Okay, you know MRSA and E. coli 0157H7 and Vibrio cholerae. All of these, they express their virulent factors through quorum sensing. Same principle with how you express light. Okay, so how do we combat this? Okay. So one is the what you call the rational drug design. Meaning, if this is the original signal, okay, gagawa ka ng analog niya na kabukha niya. Okay? Remember, the signal enters a specific receptor. Okay? So you make a dummy signal so that the receptor will be plugged. Okay? This signal will only plug the receptor, but since it is not the real signal, it cannot enter the cell. Hence, it cannot trigger the expression of your virulent factors. Okay. Yeah. So, konting quiz. If this is the signal, ano yung analog na pipiliin mo among these synthetic molecules? Okay. Dito ba? Dito ba? Dito ba? Dito ba? Dito ba? Remember that this receptor is, what's that? This receptor is specific. This signal is specific to a receptor. So the answer is actually this one. Okay. Yeah. So yeah, this is the original signal, and this is your analog. So this one will compete with this one, it will lodge here, and so you plug the communication. Okay, so that quorum sensing circuit from the signal to the receptor to the DNA to the action is cut kasi nabara lang siya dito. Okay, hindi ito makalodge, pero ito nandito siya. Okay, itong signal na to ng pathogenic bacterium. Okay, hindi na siya makaano kasi binara siya nito. Okay, yeah. So this is the normal scenario. If you have a virulent bacteria, marami sila, and they are pathogenic. And if you inject it into a mouse, it will die. Okay. But once you have, so this is the design. This is the normal situation. Pag high cell density, pathogenic sila. But kahit na pathogenic sila at high cell density, ito na yung analog na inad mo. Okay. So, na-disrupt yung circuit or yung communication. So, it seems like parang konti lang sila, ganon, kasi napaplug. Kasi konti lang yung mga autoinducers na nakaka-lodge doon sa receptor. Okay? So, nagiging non-pathogenic siya. Okay? So, it's a beautiful design. So, kahit na-infect ka, but if you have that analogs with you, those synthetic analogs with you, it becomes non-pathogenic. So, it's like going to a war, you don't kill the pathogens. You get rid of their ammunition. And that way, they will not mutate. Again, you are not applying selective pressure here. You are not, because you are not killing them. You are just muting them, okay, to become pathogenic bacteria. Okay. So yeah, so this is your, ito yung ulit yung drug candidate, yung analog. Yeah. So if you inject it into a mouse, it is alive pa rin. Okay. So you get rid of the virulence or the pathogenicity kahit ma-infect ka, and they will not become resistant because you did not kill them. Okay. Yeah. So the other method, ito, number one, ito yung pinag-usapan natin, the blocking of the signal reception. And of course, the other one is the enzymatic degradation of the signal molecule. Yung diretso mong i-degrade yung mga autoinducers, yung mga signal, yung mga bilog na red, para hindi na sila makapasok doon sa receptor. Hence, walang expression ng virulent factors. And these enzymes are called your lactonase and acylase. And the genes expressing this were actually inserted into a vineyard which were infested with this bacteria, which actually have quorum sensing mechanisms to produce biofilm. So, nalugi yung vineyard because of this bacterial infestation. But when they put so GMO to, naglagay sila ng those genes for those enzymes, then it got rid of this bacteria. So buhay ulit yung vineyard business. Okay? So may trabaho na naman, yung mga nagtatrabaho dyan. It's a multi-million dollar business. So, but papatayin lang pala ng isang bacteria, ng mga bacteria. Okay? So they applied this also in tobacco leaves and potatoes. Okay. Let's go, ano naman ang, sa Pilipinas, ano naman ang application na ano naman ang nangyayari na sa bioluminescence. Okay, so we are still in a very infantile, very early stage of bioluminescent studies. Thank you to all my thesis students who have worked enthusiastically with me. And maganda sa Pilipinas because we are all, we are archipelagic, we are surrounded by water, so we have rich aquatic resources. And the biodiversity is just amazing. So if you want to, if you cannot scuba dive, takot ka sa tubig, but you want to know the biodiversity, go to the fish market. Okay? So ako, every time I go to the fish market, I don't just think anymore of how I can cook it, but I'm always wondering, bioluminescent kaya yan? May mga bacterial symbionts kaya yan? Ano kaya yung mga ano? And that actually fueled our researches in the past few years. Yan. So ang bioluminescence is done is through yung maraming yung, you remember the Hawaiian bobtail squid? So meron siyang symbiont. So marami tayong klase ng squid sa Pilipinas. So I thought, meron bang bioluminescent bacteria yung mga yan? So ang kagandahan sa amin, pag nagsasampling kami, lulutuin na rin namin because we do not have permit to go to the wild and collect samples, but we go to the fish market. Okay. And what are our findings? So, maraming pusit sa Pilipinas. So, curious, ba't din natin sila aralin? So, our first study was subukan naman natin yung octopus. Maliit lang yan, just like your thumb. And consistently, we got bioluminescent bacteria from their head. Okay? Yan, and we identified them as photobacterium. So, and ito, iba-ibang klase ng squid. So sabi ko, subukan naman natin yung mga pusit kasi marami rin tayo niyan. Okay? So, yan. At marami sa kanila, sa ating mga pusit at pugita, marami silang bioluminescent bacteria. Pero, yan. By serendipity, we found that may mga jellyfish na bioluminescent. And I'm telling you, it is quite a spectacle. We saw this firsthand sa dagat, a school of them floating and lighting up in different depths of the water. It's just it's like a Disney movie. Ang ganda talaga, and I cried when I saw them. Okay, and not just this, a school of fish which pass underneath our boat, glowing. So ganyan kaganda ang ating mga ang ating resources. Okay, so we did have isolate bioluminescent bacteria from those organisms, marine organisms. Okay, so the question is, do they have light organs like yung Hawaiian bobtail squid? So we have a set of students, ito sila, thanks to them, and they found out na itong pusit puti, many local pusits, they dissected them. Yan. And eto, nakita nila na they only found bioluminescent bacteria within these structures. Okay? Yan, sa ventral part din. And these structures are not found in other types of squid. So they also have a morphological identification of the squid, Luligo chinensis, at yung ibang other types ng squid, wala silang part na yan at wala silang ma-isolate na bioluminescent bacteria. So for now, this is up for review. We're claiming it na ito yung mga pusit na they have light organ, and maraming ang bioluminescent bacteria yan. Magandang i-metagenomic study rin yan. Okay? Ayan sila, another type. Bale, tatlo yung nakita nila na merong light organ. Okay? Ito naman yung walang light organ. Ito yung kaibahan. At walang exact yan. At wala kaming makitang dito. Yan yung kaibahan. Yan. And these are the bioluminescent bacteria which they have isolated from the light organ. And they cultured them. Yan, umiilaw sila. Okay. Ito naman, so ito naman we tried to see, meron din silang light organ, itong mga pusit na to. This is in Ilocos, kasi mukha Kim Mandela, parang kandila kasi, yun ang local name. Yung mga bacteria, sadly, we found them to be ampicillin and penicillin resistant. Okay? Kahit na nanduod sila sa malayo sa shore, sa open water. So that's the smoking gun in this study. Okay? Yan, ito yung mga light organ na nakita doon. And then we also, sa aquaculture fish, we also sadly isolated bioluminescent bacteria which are multiple drug resistant. So ito yung mga isolates. So resistant ito sa tetracycline at saka sa ampicillin. At itong mga strains na to, resistant sila sa ampicillin at polymyxin. Okay. So kinakain natin itong mga bangus na to, grill natin at pati yung tilapia. So let's be careful. But again, this is up for review. Baka ayaw nyo nang kumain ng bangus at tilapia. Okay? So yung oysters and mussels then, we have isolated bioluminescent bacteria. And ito yung mga data na, data, pero matulungkot ka rin kasi ampicillin resistant sila. Yung mga 13 strains na isolate namin from mussels and oysters. They are farmed. So these are all the other fish na associated with bioluminescent bacteria. Itong mga bacteria na to, they can excrete anti-quorum sensing, and this was published. Okay, anti-quorum sensing molecules. It was from Taal when I was in ADMU. Hello to Ateneo, to my Ateneo students. And ito, alam niyo yung alamang, meron, dito sila hina-harvest yung mga alamang sa Cagayan, sa Apari. This is an estuary. What is so sad here is there is black sand mining going on around here at yung effluents nag-flow dyan kung nasaan yung mga doon hina-harvest yung mga alamang. And we found na yung zinc dyan is 3.5 ppm. At ang toxic level is 0.3. Can you imagine yung toxicity ng water na to? And yet, through the years na matagal na yung black sand mining dyan, nag-exist pa rin yung mga alamang. So, we isolated with tong alamang na to. Bakit kami na sa alamang na yan? Because they come in schools. It's like a milky cloud, meaning pag nandun sila sa river, meaning they are also bioluminescent. And we isolated the bioluminescent bacteria. Yan, and we found na sila, they are resistant to zinc. So pwede silang gamit up to 6 ppm. So pwede silang gamitin as bioremediating agents. Okay, in zinc contaminated water. So the next experiment, we should do experiments na they can remove zinc. Okay, kasi they can survive it or they use it for their metabolism. Okay, so one of the most astonishing encounters we had was we went to Ilocos and we saw these bioluminescent mushrooms. Okay, and this is so surprising. It's so nakakatawa ang mga field trips namin because hinahabol namin bago mag-umaga habang gabi. Sige na. While we were looking for the mushrooms, alam niyo kung anong nakita namin? The forest floor, which was filled with litter, was glowing. Naglo-glow yung forest floor filled with litter. At ditong mga litter na to, sila yung mga naglo-glow. It was such a spectacle. So, this is lights on. This is lights off. Okay? So, bakit sila naglo-glow? Okay? This is lights on. This is lights off. Okay? So, may mga fungi dyan na naka-embed. Okay? Or on the surface, kaya pala sila umiilaw. So, and we tried isolating them. Ito yung mga na-isolate naming fungi, but we failed to culture them in vitro na umiilaw. So, we don't know kung sila nga itong mga fungi na umiilaw or maybe we have to improve the nutrients of our in vitro medium, our synthetic medium para umilaw sila. There's a lot to study. Okay? Ayan sila, nagwagi naman kami sa iba, pero not all. Okay? Lights on, lights off. Okay? We were able to culture them in vitro. Pero marami sila, so yan lang yung a few. So I have students who, ano ang function yan? Ba't umiilaw sila? They say that they can attract insects so that yung spores ng mushroom parang pollination i-spread ng mga insects. So we have these two setups, itong synthetic na light at walang light. So and then binilang namin after these hours kung may mga may mga insects kang matatrap diyan. And ito nga pag with light, ito yung mga insects na na-attract. Pag no light, ito. So it seems like may phototactic kwan ang mga insects nga. Pero again, this needs to be followed up to have a concrete conclusion. Okay. Okay, I would like to end up. That's all I have now. I would like to end up that there is with the saying that this is a quote from our biodiversity expert. This is a single species that is responsible for the COVID-19 pandemic, and that's us. Okay, because my heart goes to, I worry because before we can study these organisms, these bioluminescent organisms, they're in the waters, in the forest, everywhere, but because of human anthropogenic activities, we encroach into their natural habitat. I worry that mawawala sila, and I've seen this firsthand. Like here in Baguio, recently, di ba may mga orca na nakita sa Bohol? And while I was preparing these slides, and may nakita akong mga fireflies sa kwarto ko, and then sa kitchen namin, and then sa kwarto ng nanay ko. And this rarely happens. Ngayong may lockdown lang, tsaka nangyayari yun. So maybe I was thinking maybe the environment is now improving. Okay? So the best action we can take is to protect our natural resources and the environment. And I would like to end with a short clip. Okay, this is bioluminescence from the from the forest, from David Attenborough's BBC video. When we were looking for mushrooms, the locals said ang dami-daming mushrooms doon sa forest nila na umiilaw na it was a common thing for them. Pero yung part ng Ilocos na yun na pinuntahan namin, it's now a tourist spot and at marami ng resorts. And sadly, sabi nung guide namin, wala na yung mga bioluminescent mushroom. Actually, dalawa na lang nung nakita namin. Noon, ang dami nila. So, this is the fireflies. They are blinking in unison, in synchronous ways. And until now, scientists don't know why they blink synchronously. Okay? But if we too much development, mawawala tong forest na to, mawawala yung mga microorganisms na yan, and fireflies. And wala na. I hope we really take care of our environment. So thank you, stay well, and stay safe.
Thank you very much, Dr. Kalugay, for that very enlightening talk. Now, we now move on to the third speaker, but before that, please take note of some of announcements from the.
So the announcements are being flashed on your screens from time to time. And then we will again be flashing these announcements later on, okay? So perhaps we'll be moving on to our last speaker, but definitely not the least. Of course, Mr. Angel June Barnes. So June finished Bachelor of Science in Biology, major in Microbiology from the University of the Philippines, Los Baños. He also pursued a Master of Science degree in Microbiology with minor in Biochemistry from the same institution. So his professional experience include 9 years of teaching at the Microbiology Division of the Institute of Biological Sciences at UPLB. He also served as the Microbiology Laboratory Manager of Dole, Philippines in Polomolok, South Cotabato. He transferred to 3M into 2007, where he started as a professional service analyst for the medical division. A year after that, he transferred to the Food Safety Department. Currently, he serves as the professional service for the national key accounts of the division. So as part of 3M, he has been invited to several local, national, and international training conferences, conventions, seminar workshops, as participant or as a speaker. His work has given him the opportunity to reach out to 3M's growing number of customers while being able to do two things he loved the most: teaching and traveling. So ladies and gentlemen, please help me in welcoming June Barnes, our last speaker for today's webinar.
Thank you very much, Dr. Len. I'm really very happy to be a part of this PSM webinar series. To all the participants who are logged in, good morning to all of you. Once again, I am June Barnes. I'm also a microbiologist, and currently I am working as the professional service for 3M Philippines Food Safety. I really feel so privileged to be speaking alongside Dr. Grace Banaay and Dr. Ronnie Kalugay for today's session. And the fact is, the presentation of Dr. Kalugay is actually a very fitting prelude to my presentation this morning, because I will be focusing on some of the very practical applications of ATP bioluminescence technology when it comes to microbial testing as used in food industry. You will notice that the applications are very simple, but the value that they provide to the food industry is really very, very important. Just very briefly, right after teaching in UP Los Baños, I worked very closely with the food industry. And for the past 15 years, if there's one source of joy in being part of the food industry, that is I get to see how the foods that I buy gets prepared. I think that's a privilege I have. And I'm also very happy to witness how the food industry here in the Philippines has grown. And it has really grown a lot. Some of the big industries we have would include fruits and vegetables. We are a top manufacturer of desiccated coconut and dried mangoes. We're also very big with our pineapple products. We also have a big meat and poultry industry, as well as fish and marine and dairy industries. And I'm also happy that we are growing our ready-to-eat market. I think it's a sign that more and more people are becoming a lot busy, and we are depending on manufacturers of ready-to-eat food products. We also have a good bakery, beverage, and snack industries. And as you can see in the slide, there's really a wide variety of products which are manufactured here in the Philippines, whether it's for local consumption or for export markets. But despite the diversity in the products that are manufactured by these companies, there are actually two things that are shared by all of them. That is the need for them to ensure quality of their food products, and the need for them to ensure safety of the food products before they are sold in the market. And for microbiologists like all of us who are logged in today, I'm sure that all of you know that microbial testing is one of the tests that needs to be done in order for these food manufacturers to know the number of microorganisms in their samples, if they are within standards, and in order for them to detect the presence of pathogenic microorganisms, which should be absent in their food products. Now, when it comes to microbial testing, they are not limited only to raw materials and end products, but they are also applied now widely for environmental samples. Now, I'm really very happy that for the last 10 years, when it comes to microbial testing, I would have to say that more and more food manufacturing companies are really looking now for ways in order to simplify their process of microbial testing. And they also want to significantly reduce the amount of time needed for getting the results without really sacrificing the quality of the test results. The fact is, whenever you go to food manufacturing companies and you ask the analysts in their microbiology laboratory, what is it that they are looking for now? A majority of them would say that they really need speed. The faster, the better. And this is really clearly indicated by the growing demand for alternative rapid methods for microbial testing. And I really believe that one of the reasons for this is because a lot of managers, a lot of owners, even laboratory managers and analysts are now seeing the value of getting the results fast. Because once they get the results fast, that means that they can release the product sooner. And with tough competition in the market now, food manufacturing companies really need to make sure that their products are readily available. Otherwise, there's always another brand that consumers can purchase. Think about coffee. When I was still very young, probably the two most popular brands would be Nescafe and Blend 45. But nowadays, if you think about coffee, aside from these two brands, you have a lot of local and imported brands. So imagine if you are not able to release your products early because you have to wait for the micro results. That means that there's always another brand that your customer can buy. And of course, as a food manufacturer, you don't want to lose your customers. Now, for food manufacturing companies which are producing products with short shelf life, getting the results fast would allow them to maximize the shelf life of the product. Imagine if I am manufacturing bread, which has a shelf life of only about 7 days, and I need to wait for 5 days for my yeast and molds. That only means one thing. By the time I have my YM counts, I only have 2 days to market my product. And that's, of course, not very good for my business. And lastly, faster results would also allow food companies to have a better control of the manufacturing process. Just try to imagine having salmonella in your plant, and you found out about it 4 to 5 days after testing. That would only mean one thing. For the past 5 days, while you are waiting for the results, that means that you have not done anything to control the organism early on, simply because you did not know that salmonella is actually in your plant. That would be a big loss for the company because there's a very big possibility that the other lots that will be manufactured would be contaminated with salmonella. And because of this, because of the facts that I have mentioned to you, more and more food companies now are really looking for innovative technologies, alternative methods that would allow them to detect, quantify, and identify microorganisms, again, not only in food, but also for environmental samples, since more and more food manufacturing companies are focusing now on their environmental monitoring program. Now, I'm really very proud to be part of 3M, and one of the reasons for this is because 3M has been one of the key players when it comes to the alternative or rapid method platform. And 3M is also considered to be one of the most innovative companies in the world. And I will be sharing with you a secret. Our secret basically lies on what I am showing to you now. So if in chemistry we have our periodic table of elements, in 3M, we do have our 51 core technology platforms. And basically what we do is to combine one technology with another in order for us to come up with a new product that would meet market needs. These technologies are divided into materials, processing, capabilities, digital, and of course, applications. Now, just to give you an idea how we do it, let's have one example. If there's one thing that 3M is really known for, it would have to be films. You know our Scotch tape a lot. And if you're using 3M cartons, these are films. So how do we apply now the film technology for microbial testing? It's very simple. Basically, what we did was to combine films, the same ones we use for our medical tapes, with other technologies like adhesives, biomaterials, polymer processing, and etc. And such combination actually led to the creation of what is popularly known as the 3M Petrifilm, which has been utilized by laboratories around the world for indicator testing for the past 36 years. Interesting, isn't it? But if you are going to ask me now, which among the 51 core technologies is my personal favorite, it would have to be light management. And the reason is because this is where ATP bioluminescence technology is included. Sir Kalugay talked about light. And when you think about light, don't you find it amazing that when God said, "Let there be light," he meant it not only for us to have a beautiful view of the sunset in Manila Bay, or for us to have a brighter celebration of Christmas. When God said, "Let there be light," he actually meant it also for us to have safer foods. And that is exactly what I want to highlight in this presentation. How can ATP bioluminescence technology be able to bring light to food safety? I was fascinated by Dr. Kalugay's presentation earlier because he featured a lot of organisms which are capable of producing light. And he mentioned that some organisms like algae, jellyfish, some fish, and even bacteria can emit light because of chemical reactions in their body. I remember when I was taking my master's in UPLB, the organism, the bacterium that was assigned to me was Photobacterium leothi, which I got from squid. And it would actually emit light. Very, very interesting. Now, this chemical reaction is, of course, called bioluminescence. But for me, one of the most fascinating displays of light by a living organism would have to be that of fireflies. In fireflies, bioluminescence occurs in specialized light-emitting organs, which can be found in their lower abdomen. And this is made possible by an enzyme, which is called luciferase, in the presence of luciferin. Now, the reaction that is catalyzed by the luciferase enzyme was first studied in the North American firefly called Photinus spiralis. And just like anything that lights up, in order for bioluminescence to be displayed, you need one very important component, and that is an energy source. And that energy is supplied by ATP. So this reaction was first described in 1947. So once ATP reacts with the luciferase enzyme, that is when you're able to produce light. And that light is expressed in terms of RLU, which stands for relative light units. You know ATP very well. Uh, I'm sure that all of you are very familiar with this one because this is our energy molecule. So all living organisms would have it because that is where we store our energy. Now, this figure here would show to you the chemical reaction that is involved. Basically, what happens is when oxygen combines with ATP, and of course, you need to have your substrate, which is the chemical luciferin, in the presence of luciferase, which is a bioluminescent enzyme. That is when light is produced. And take note that there is a direct relationship between the amount of light that is produced with the amount of ATP that is supplied. So that only means one thing. The higher the levels of ATP that is supplied, this would generate higher values of relative light unit. Now, in 3M, in the food industry, we have utilized ATP bioluminescence in a wide variety of very practical applications. And let me take this time to focus on some of these applications. You can use ATP bioluminescence technology now for hygiene monitoring. It's also used for UHT processed end product testing. And one of the newer applications of ATP bioluminescence would have to be for pathogen testing. So let's discuss now in details how this technology has been utilized for these different applications, starting with hygiene monitoring. I'm really very happy that its application for hygiene monitoring was developed because it's really one of the practical applications of this technology. Of course, if you're working in a food manufacturing company, including hotels and restaurants, isama na po natin yung mga hospitals, cleaning is a very important activity. So doing proper cleaning and sanitation, they are necessary in order to ensure safety of the products that they are manufacturing. Now, in food manufacturing companies, they normally follow very strict protocols when it comes to qualifying the raw materials and water that they are using, as well as having really strict procedures for clean processing and packaging of food products. However, even though all of this are perfect, it is still not enough. Why? Because if the equipment that is being used is dirty, whatever is present on the equipment, it may actually go to the food product. And as reported during the International Association for Food Protection Annual Conference that was held in Rome in 2007, they said that whatever it is that you have on your equipment, there's a 70% chance that it will get into your food products. So you really have to do proper cleaning in order for you to get rid of these dirt. The reason why it's really very risky because some of the product residues may contain allergens and you don't want introduction of these allergens into your food product. Furthermore, whatever product residue is left on the equipment, that may serve as a breeding ground for microorganisms which may eventually cause spoilage or food poisoning. And that is why, aside from focusing on the quality of the raw materials and water, plus following process and packaging strictly, it's really important that hygiene is perfect too in order to prevent cross-contamination and cross-contact. So based on our discussion, I think I'm very clear in pointing out that cleaning is very critical. And since it's very critical, it's really important that we go beyond the surface when it comes to inspecting cleanliness. Because the reality is we cannot see every health hazard that you have on your equipment or plant. So how exactly do you see clean? Now, in the past, many food manufacturing companies would just rely on visual assessment. The nice thing about it is that it's really cheap. You just need your eyes. And it's also very fast and fairly simple. I mean, the moment you look on the surface, based on your understanding of what clean is, you can immediately decide if the equipment is clean or not. But of course, it comes with a downside, and that is is a very subjective approach. I mean, the way you see clean is very different from the way I see clean. And it's also difficult to set a standard. Plus the fact that it's not a very sensitive method because what we can see is quite limited. And that is why to verify cleanliness, we really need to have an assessment that goes beyond using our naked eye. And that's why a lot of food manufacturing companies now would also perform swabbing and subject it to microbial testing. Now, the good thing about this approach is that you are very objective now when it comes to decision making because you normally follow a standard. For example, if a food manufacturing company has a standard of 50 CFU per 100 square centimeters, any value below that, you can conclude that the equipment is clean, and any value higher than 50 would mean that the equipment is still not clean. The challenge, however, when it comes to microbial testing is that it requires a long time for lab results to be generated. And it's really more retrospective. Parang naglinis ka ngayon, nag-swab ka, malalaman mo yung results 2 days after. So what if it's dirty? Hindi naman pwedeng 2 days after, pupunta ka sa planta, galit na galit ka, pag sasampalin mo yung mga tauhan mo para sabihin 2 days ago, ang dumi ng planta natin. It's important because you know the number of microorganisms on the equipment, but you were not able to address the issue prior to production. And because of this, what food companies really need now is a reliable, complete system that would allow them to manage their hygiene monitoring process without the wait. And this is where ATP testing comes in. Now, the main advantage in ATP testing is that you can get the results really fast. How fast? Well, less than a minute. You can already get the results. And based on that result, you can decide if the equipment is clean or not. If your standard, for example, is 150 and a value below that is considered clean and therefore production can already begin. And any value higher than 150, that means the equipment is still not clean and therefore you can ask your team to re-clean first prior to starting your production. And you now are very objective when it comes to your decision making. So in this application of ATP bioluminescence technology for hygiene monitoring, you make use of swabs. And these swabs would contain extractants. These extractants are really important so that they can cause or aid in lysis of cells, including microorganisms, so that the ATP can be released. Now that ATP is then mixed up with the luciferase enzyme, which is contained in the cuvette. So once you activate by clicking, that is when light will be generated. And that light now will be expressed in terms of relative light unit. Now, the instrument that you use for getting the reading is what we call the luminometer. So for food facilities, after they have done cleaning, what they want is to get an RLU level that is as low as possible, because that is a sign that they have low levels of ATP. And low levels of ATP would mean that they have low levels of contamination on the surface. Very simple application, and yet it's really very helpful. And I'm really very happy that here in the Philippines, the use of ATP technology is really widely appreciated. And they have been used in a lot of hotels and restaurants now, especially now that we have our COVID-19 pandemic. They are really into hygiene monitoring now, making use of ATP bioluminescence technology. The nice thing about ATP, the reason why it's regarded as an excellent hygiene indicator, is because you don't take into account the direct risks only. Yes, it can measure microorganisms, which is your direct risk because they can cause food poisoning and spoilage. But I think another value that ATP technology has is that it can also measure ATP coming from organic residues. And these organic residues are actually indirect risks because the moment they are left on the surface, they may encourage microbial growth. So it's really important that we measure the total dirt which is present on the surface prior to deciding if production will continue or not. Now, aside from its use in the food industry, I'm happy to share that there are ATP technology is widely used now even in hospitals in other countries in order for them to assess cleaning and sanitation of critical areas, like, for example, the operating rooms. As you can see here, prior to cleaning, the OR surface would have reading of greater than 10,000 RLU. But after cleaning, the RLU was reduced significantly. It's now only about 347 relative light units. Now, aside from assessing cleanliness in different rooms in the hospitals, they are also they are also used now for verifying cleanliness after cleaning of endoscopes prior to sterilization. But of course, one of the most widely used applications of ATP bioluminescence technology would be for checking of cleanliness of food preparation areas. And I'm happy that in other countries, even 7-Eleven stores, like when you go to Thailand, all 7-Eleven stores there are actually being audited and checked regularly using ATP bioluminescence technology. And when you go to Japan, many of the fast food chains there, hindi ko na lang po sasabihin yung pangalan, are checked using ATP tests. Now, in many hotels, food manufacturing plants, and also hospitals, ATP tests are also widely used for checking of hand washing efficiency. As you can see on the screen, before hand washing, the reading that they got was about 104,000, more than 104,000 RLU. But after hand washing, the RLU level that was reached is only 88 relative light units. So the nice thing about this is that you can really objectively decide if your team is doing cleaning properly or if they need further cleaning prior to start of production. Now let's take a look at the second application of ATP bioluminescence technology. This is very interesting. This is used for end product testing of UHT processed beverages. Now, when this technology was first applied, it was initially intended for UHT processed dairy products like milk, flavored milk, and also dairy substitutes. But recently, this was also applied now to other UHT processed fruit juices, as well as beverages, like, for example, the UHT processed coconut water, which is now a booming business here in Southeast Asia. Now, as you know very well, once you have your raw milk and once you open the coconut fruit, the coconut water, once it's exposed to the environment, would contain microorganisms. And some of these microorganisms would may cause spoilage, and some of them may be pathogenic. And that is why it's important to treat these products in order to render them safe. With traditional pasteurization, you're able to render the product safe by killing the pathogenic microorganisms, but still some spoilage microorganisms would remain. And that is why refrigeration is necessary during storage of these products. Now, an alternative method is UHT, which renders the product commercially sterile. However, as much as we wanted to make the process perfect all the time, and I believe participants here who are part of a company manufacturing UHT would agree that there are times wherein there may be issues, particularly during filling and packaging, which may introduce contaminants into the product. And this is where the challenge now comes in, because UHT products are stored under ambient conditions. So there's the condition is perfect for growing the organism, and eventually issues like bulging may happen. Now, when these UHT processed beverages are produced, they are not tested immediately. They are, unlike other food products, once you produce, you can submit them to the laboratory for immediate testing. But for UHT processed beverages, we don't do that. Because there's a very big chance of missing the contaminant. Imagine if you only have one cell, one contaminant in a product which is 1 liter or 333 milliliters of the product. Finding that single contaminant would be very, very hard. And even if you take a lot of samples out of the container, yung tipo ang dami mong plinate, it would still be random luck to find the microbe. And because of this, it is really important for UHT processed beverages to undergo first a pre-enrichment stage. And this is normally done for a long time, about 5 to 7 days, depending on the company. And this is done at 35 and 55 degrees Celsius. And this step is really very important in order to give time for the contaminant to multiply. Since the number of microorganisms increased significantly, you're making now the test a lot more sensitive because there's a larger number of bacteria in the carton. And because of this, you will be able to decide better whether the product is okay or not okay before you sell it. And this is now where the issue comes in. For food manufacturing companies who are producing UHT processed beverages, and if they're using traditional agar, it's a challenge because they already allotted time for pre-incubation. Imagine 5 to 7 days. And then you will plate. And therefore, you will have a second incubation, which will take about 2 to 3 days. You need this in order for the microorganisms to show up. Now, this would mean a lot of costs for the company for warehousing and also a lot of delays in generating sales for the company. Good thing, we have our ATP technology, which is very helpful because ATP would allow you to eliminate the second incubation. And at the same time, you can actually shorten the pre-enrichment period. How short? Now, based on existing customers, they can shorten the pre-enrichment from about 5 to 7 days to just 2 to 3 days. And the actual ATP test would only take less than an hour. That would mean instead of holding your products in the warehouse for about 10 days, you just need to hold your products for 3 days. And that's a lot of time savings for the company, which has been using ATP bioluminescence technology for this application. Now, this is a very interesting question that we often receive from customers and from people who want to know more about this application of ATP bioluminescence technology. They would ask, how is the system able to differentiate the ATP coming from the actual food sample and the ATP which is coming from the contaminating microorganisms? Remember that your samples, milk, for example, and coconut water, these are biological fluids, and therefore they would also contain ATP. How do we differentiate now the ATP from the product and the ATP coming from the contaminating microorganism? Now, the answer lies on the use of an enzyme called ATPase, which would allow you to remove the product ATP. Now, in order for me to illustrate that further, let's take a look at this figure. Imagine you have two samples, one which is clean, that means no contamination, and you have another product which is contaminated. So as you can see in the clean sample, you only have one source of ATP, and that would be the product. In the contaminated sample, you have two sources of ATP. You have your microorganism and you have the product itself. Now, during the UHT assay, what is introduced first into the sample is the enzyme ATPase. Now, during the incubation period, that is when ATPase will start removing the ATP coming from the sample. So you're basically left with just the ATP coming from the contaminating microorganisms in the second well. So how are you able to release now the ATP coming from the contaminating microorganisms? This is where the extractant comes in. Basically, what it does is it would lyse the cells so that you can release the ATP. Lastly, you introduce the luciferase enzyme, and because in your clean sample you don't have any source of ATP, once you get the reading, it will be very, very low. And in the case of the contaminated sample, since you have your ATP from the contaminating microorganism, the RLU level will be significantly high. Again, another simple application, but it's very useful for the food industry. Now, the last application that I'm going to focus on would be for pathogen detection. And in 3M, I'm really very happy that we have innovatively combined ATP bioluminescence detection with a molecular method called LAMP, or loop-mediated isothermal DNA amplification. This is this is really an amazing combination of two robust technologies that would offer specificity and sensitivity that a lot of food microbiology laboratories want in a pathogen test solution. It's also a lot faster, very simple, and at the same time cost effective. Now, just to show to you how it works, let's take a look at this figure. So in isothermal DNA amplification, you make use of multiple primers. And these primers are very important for specificity. They would recognize specific or distinct regions on the DNA of your target organism. Now, instead of using Taq polymerase, we made use of BST polymerase, and the combination of the polymerase and primers would provide continuous and rapid amplification. Now, during the production or amplification of the DNA, you produce a lot of pyrophosphates, which are then allowed to react with a molecule called adenosine phosphosulfate. In the presence of ATP sulfurylase, these are combined, allowing you to create ATP. Once you supply luciferase enzyme, that is when you produce light. So if you're going to simplify the process, it simply says that if you have light, that means you have amplification because you are positive for the organism. And absence of light means you are negative for the target organism. Now, just a simple illustration. Imagine if you have your target DNA and you want to amplify it. Again, you would need your primer, you would need your polymerase, you would also need your sulfurylase enzyme so that you have a glue that would bind the pyrophosphate to your adenosine phosphosulfate. Of course, since we are talking about ATP bioluminescence technology, you need to have your luciferase enzyme plus the bases. Now, in isothermal DNA amplification, you only use one temperature. Our amplification temperature is 60 degrees Celsius, and that's when the primer would look for its partner on the target DNA. The polymerase will bind to it, and that will now start the amplification process. Now, as I've mentioned earlier, amplification will lead to production of pyrophosphate. Since you have adenosine phosphosulfate, sulfurylase will convert those two to ATP, which will now react with the luciferase enzyme. And that is your signal that you have your amplification. So how will you know you have the signal? Well, you make use of a software, and a peak like this would tell you that you are positive, which means that the target organism is there, while a flat line like this would show that you are negative for the target organism. Now, lastly, I just want to highlight the advantages in the use of ATP bioluminescence technology for pathogen testing. One of the major advantages is that it would allow you to have reporting of the DNA amplification of the target organism in real time. And as I've shown earlier, you basically have a two-step enzymatic process here, wherein you have your pyrophosphate combining with adenosine phosphate so that you have production of ATP, which then reacts with luciferase to generate light. Now, another advantage is that you don't have to use any more high-cost excitation sources. You don't have to use fluorescent dyes and fluorescent filters and detectors, and it would significantly cut the cost for your pathogen testing. ATP bioluminescence technology has also been reported to be a very robust and reliable detection method for molecular testing because it can resist sample interference. One issue that has been encountered with fluorescence detection, which is used in many PCR-based systems, is that it can be subject to natural interference by natural fluorescence of some food samples and enrichment media. So if your product is highly colored, like turmeric, there's a chance for you to have false positives. And lastly, the use of ATP bioluminescence technology in our system that we have developed has really allowed us to just have one protocol for all pathogen tests. So imagine having 8 targets, and you just need one protocol for all of them. And you only need a single enrichment, and you only do 2 transfers from your enriched media going to your lysis tube to get the DNA. And this DNA is then transferred to your reagent tubes, which contains all of the reagents. Once you place the tubes in your instrument, that is when amplification happens. So one protocol is applicable to all the pathogens that can be detected by the system. And because of this, if you, for example, need to test for multiple organisms, you can actually do testing simultaneously. And that's a major advantage in many food microbiology laboratories. Now, in summary, based on the applications that I have shared to you this morning, I really hope that I was able to show to you how powerful and useful ATP bioluminescence technology is for microbial testing. And as I have shared, ATP bioluminescence can allow your laboratory to have faster, easier to use, reliable, and cost-effective means of testing for the food industry. And currently, these are the applications for ATP bioluminescence for hygiene monitoring, UHT product testing, and pathogen testing. Now, just in case you have questions about this technology and you want to know more about how we can help you, please feel free to contact me at this number. And you may also email me at jbarnes@3m.com. Isang magandang hapon po sa inyong lahat at maraming salamat sa inyong lahat.
Thank you very much, Sir June. That was a very, parang ang ganda, no? Continuation talaga siya, no?
Yes.
Theory na no,
Yes.
shinare ni Ronnie, no? Thank you very much. Sir Noel, do we have a question for
Yes,
Sir
I
June?
think we have for the open forum. Would like to inform the participants. Those with questions, they have been answered.
Sir Noel?
Sir Noel, I think some of the questions kasi for Dr. Banaay and Dr. Kalugay, they started answering in the chat box already. So we have now just a question for Sir June. Yan. So sandali i-open ko ang kodigo ko. Yan. So Sir June, this is your question, no? This is a question from Jasmine Velo. Jasmine, wave ka kung anong institution mo you didn't put. And also,
Hi, Jasmine.
A connecting question then from Sir Hil Penular of UP Diliman. No? So the question goes like this. Does ATP test count only the overall bacteria present in the surface or the sample, since it lyses all the cells in the swab? No? And I think this is also connected with that. Most light-based and bioluminescence-based detection methods are affected by interference coming from organic compounds or, say, detergents. No? So how does your 3M ATP bioluminescence-based technology address that concern? No? Sir June?
Okay. That is actually a very good question. And if you will notice, I really actually decided not to focus too much on our product, but I really want to focus a lot more on the technology. And when I say that, I am speaking on behalf of all the manufacturers of luminometers and ATP systems used for that application.
With that technology.
Yes.
Okay.
So let me concentrate first on the first question, which is it specific only for the microorganisms which can be found on the surface? The answer is no. And that is the reason why ATP tests are considered to be biological tests and not microbiological testing. Because remember that any product residue, plant-based or blood, for example, from animals would have ATP in them. And once you use the swab, that will not be able to differentiate, including the luminometer, will not be able to differentiate which ATP is coming from your microorganism and which one is coming from your organic residues or whatever that cell is. So it has to be clear to everyone that what you are measuring here would be the total dirt. And when you talk about ATP tests, it actually answers just one question: is the equipment clean? So when you talk about clean here, it can be when you talk about not being clean, it may be because of microorganisms, or it may be because of the presence of other
I
cells
agree.
from animals or plants or whatever that source is. Now, with regards to the second application, organic residues, I really think that it's best for you to consult the suppliers, because I do believe that different suppliers would have their own studies when it comes to how what do you call this, detergents and how sanitizers can affect the performance of their swabs and luminometers. In the case of 3M, it's something that we have done extensively, because we do know that there's a wide variety of disinfectants which are being used in the food industry. And there's a list that I can provide to you. And the nice thing is that as long as these disinfectants are used within the level that's approved by the supplier of those disinfectants, we have tested that there is no effect. But it's still best for us to have some sort of like verification, because we want to to really have a solid information that quenching, that's the term that we use, is not happening when you use the ATP system. I hope that answers the question. Ma'am Jasmine, I hope I answered your question.
Okay. So Jasmine and Hill, I hope your questions were all clear, no? So that's practically the key thing there, no? So parang if this is the particular methodology, no, you have to use it based on saan nga ba yung decision, gagamit ko ba to sa ganitong decision or not, no? So that's basically the most important thing to consider, right, sir? Because
Yes.
I'm sure all technologies will be superb, no? But of course you have to choose, is this the applicable technology for my
Right.
needs, right? Okay. So
And
thank you very much.
the
Yeah,
truth,
yes,
mom,
sir.
Len, is that if we work with one company with multiple plants, for example, one standard, the standard of one plant is not necessarily the same standard that will be used by another plant, because again, you have to do a some sort of validation study in order for you to come up with the best limits to use.
Okay, thank you very much, Sir June. So I'm sure all of the participants here in the food industry would be very eager to learn more about that technology from you and other companies as well. No, so Sir Noel, are you here now? I think we are moving forward. Dito na ba si Sir
Okay,
Noel?
so thank you very much, Surgeon Barnes. I'll just send through email my questions.
Sige, sir. Sige, sir. Basahin ko na lang
Pinalilis
later.
ang pa-quiz sa'yo.
Okay?
Naku, kinabahan naman ako sa t-shirt ko sa Micro 150.
Okay pa. Okay, so medyo overtime na po tayo. So this time, may we call on our the chair of the webinar committee, that is to give us the closing remarks.
Hello, good afternoon, everyone. Again, good afternoon to my colleagues and friends. I would like to thank everyone for participating in the day 2 of the first ever PSM webinar. As of our latest count, we even reached 360 participants. And we even had a participant all the way from Indonesia. So thank you very much for making our first webinar series very successful. As mentioned earlier by the President, we will have more PSM webinar series as part of the new normal. We hope that you will continue to support and participate in all our future webinars. I would like to thank our PSM speakers. Yesterday, Dr. Zarate, Dr. Gos, and Engineer Gue. Today, Dr. Banaay, Dr. Gos, and my former professor in Micro 1, Sir Barnes. Again, I truly appreciate the very positive response to our call, even in a very short notice. Thank you to my PSM colleagues, the support, especially Dr. Villegas, Dr. Sabino, Sir Vasquez, Dr. Papa, Sir Pagoso, and of course, Mr. Datul, for helping me on the technical aspect of this webinar. Again, thank you everyone, and please continue to support PSM and our new PSM president, Professor Joel Cornista, and his board. And let's hope that we will be able to physically and safely see each other again soon. Thank you very much.
Thank you very much, Sir Christian. Now, for all our participants, thank you for attending, and for announcements, please visit the PSM website for future activities of the organization. Thank you very much, and see you in future activities. But before you leave, please take note of the link here in order for you to get your certificate. So please answer the survey. Now, please take note that the survey link is only available for 24 hours. Okay, so please take note of the link.
For the members of the board and the speakers, please stay for a picture taking.
Thank you very much, everyone.
See you in future activities.
Jordan, i-on nyo na yung camera nyo para makapag-picture taking na tayo.
Tanggalin ko na po yung share. Okay na siguro.
Baka kinokopya pa nila yung link.
Are you sure tayo na lang ba? Baka mamaya mag-joke ako.
Dali.
121 participants pa nasa loob.
Oh,
Sir,
Nasa loob.
nag-remove pa po.
And I'll request Kenneth to cut yung YouTube na rin natin.
101.
Meron pang nagpapa-admit.